Hello, everybody. If I could encourage everybody to move up to the front. Um, this is a huge room that is set up for some of the later talks that are expected to be much larger. But please move up. We want to make this as close and personal as we can. You don't have to have a table to yourself. You can sit with other people. And we'll get started in a minute. Thank you for.
It's showing your presentation. Pointing on it, then you have to move your face and you really
Oh, and you guys, we only have the two mics. So you guys. All right. Hello, everyone. We're going to go ahead and get started. Thank you. Rounds of applause already. My name is Dan Harvey. I've been working with the foundation for a long time, been on the board and been working with the Scientific Advisory Committee and our scientific community for many years. It's an honor to moderate this research panel Q&A. I want to encourage, this is supposed to be an interactive session. So I'm going to have the speakers or our panel introduce themselves a little bit. And then I have some questions to present that I think are representative of the kinds of questions that all of you might have. But please, we have two microphones to the left and the right of the stage. If you have questions that you would like to ask, please come up to the front and use one of the microphones so that everybody can hear you. This is a panel to focus on the current state of research and discuss the future programs, some of which Ipe talked about and were talked about in Becky's presentation just earlier this morning. We're here to talk about those things and what is, help you learn about what's going to be coming in the future, where we are and what's going to be happening in the next 5 to 10 years or longer. There will be talks later today that are going to talk about the current status of things that are in the clinic. The Angelman's, the ASO trials, the antisense oligonucleotide trials in particular, those are going to be covered in detail by the clinicians that are doing the trials and by the organizations that are managing those trials. So we're going to try to stay away from the clinical side of things at this point and talk about some of the other activities, the gene therapy, small molecule things, and anything else that you would like to ask on the research front. Okay. So I just want to sort of frame what we're going to focus on here, but I do want to encourage all of you to come up to the front and ask questions as we go along. And I know we already have some people ready, but give me a minute to get to you here. So what I first like to do is have our panel introduce themselves, tell you a little bit about what they've been doing. I do need to mention Ben Philpott, unfortunately, who I was very much looking forward to having on this panel. He did have to leave last night because of a family situation, so he couldn't stay to be on this panel today. But all the three of them and myself will try to fill in and address any things that Ben would have been addressing. So with that, I'll turn it over to our panel. Mark.
All right, great. Can everybody hear me? Microphone's on. My name is Mark Zilka. I'm at the University of North Carolina at Chapel Hill. I was asked to tell you all a little bit about how I got involved in Angelman syndrome research. And it is unfortunate that Ben is not here because he's one of my very close colleagues. And it really started about 20 years ago when I joined UNC. Ben was at UNC for maybe a year and a half before me. We were both new assistant professors trying to figure out what we wanted to do with our futures and our careers. And at the time, he was studying these receptors that regulate excitability in the brain. He wasn't doing any work on Angelman syndrome, but several years after he joined UNC, he, at the advice of one of his friends, started looking into UB3A and Angelman syndrome because there were potential relationships between what he was studying with these receptors and this disorder, and started coming to some of these meetings. And then he would come back to UNC. He and I would have lunch almost every day talking about science. And he just came back so energized from these meetings where he got to meet many of the families and just realized that there was so much need for treatments for this disorder. Because again, 20 years ago, the perspective was there were no ASOs, there were no gene therapies, there were really no options to turn on the UB3A gene. And so the hope was that through research that he could do in his lab, they might be able to come up with some sort of a treatment. At the time, I was, Angelman was the furthest thing from my mind. I was like very, very new, and the advice I was given when I started a lab was, three things, which was focus, focus, focus. And so I was very focused on a different area of research related to pain. But as Ben was coming to more and more of these meetings, I talked to many families, and sensory issues were coming up as one of the symptoms with Angelman syndrome. And he was often asking me to assess are there sensory issues in the Angelman model mice. After several years, I relented and was like, all right, fine, we'll check the Angelman model mice. And that led to a project that ultimately, we did find sensory abnormalities in the pain domain. We published a paper, but it was much later. But through these meetings that we would have at lunch, we started brainstorming about how could we turn on the dad's copy of UB3A. And this led to interactions with another colleague at UNC, Brian Roth. He has these huge drug libraries, thousands and thousands of drugs. And the idea was, could we take these drugs and test them one by one to find something that could turn on the paternal copy of UB3A? And at the time, people were telling us that this was just not possible. You've heard about the differentially methylated regions in the genome, and it was thought that the UB3A gene was just permanently turned off, and there was no hope for turning it on. But we were kind of naive and young. Naive and young people tend to do dumb things sometimes. And so we tried anyway. We did this drug screen. And to our surprise, we found that there was a drug from this huge library of Brian Ross that turned on the paternal copy of UB3A. You heard about this in the last talk. It's called topoisomerase inhibitors. And we were quite shocked because these are chemotherapeutics that are used to treat cancer, yet somehow in our neurons, they were turning on UB3A. And so we wound up submitting grants to Angelman Syndrome Foundation and elsewhere, and we were very fortunate to be funded to pursue this line of research. And ultimately, we found that these drugs do have side effects that really make them difficult to use for Angelman. But this really highlighted to the field that the paternal copy of UB3A can be turned on. And then this led others like Art Boudette's lab and then a whole host of things that we can talk about today to essentially stop the stop, find other ways to specifically target this UB3A antisense transcript to turn on UB3A. And that's what my lab has been focusing heavily on now to do this with genome editor-based therapeutics. So I'll turn this over to Ipe.
Thank you. I'll keep it short since I already spoke for 40 minutes earlier this morning. So my name is Ipe Elgersma. I work at the Erasmus Medical Center at Rotterdam. I'm the scientific director of the Encore Expertise Center, where all the Angelman kids go. And this center has been already founded in 2010. So we have seen by now over 180 children with Angelman syndrome. And actually, my work on Angelman syndrome started 25 years ago. I had a favorite protein called CAM kinase that I had been studying in my postdoc for 4 years. And then it turned out to be that this protein was affected in brains of mice with Angelman syndrome. And I got really intrigued how that could possibly be. And by working on these mice, I got more intrigued in Angelman syndrome by itself and what that protein is actually really doing, the UBCA protein. And so, I started working on the Angelman mice and have done so since almost 25 years now.
Hello, my name is Ryan Butler, and I am in awe of the two gentlemen up here with me. Basically, how it all started for me was about 6 years ago. I was working at UNC Chapel Hill, same place as Dr. Zilka and Dr. Phil Putt, who's not with us. I was working as a postdoc, and my wife was working for Dr. Stephen Gray, one of the world's leading gene therapy experts. And one day he calls her into his office and asks her if he wants to move to Texas to help him start a gene therapy center there. And she told me that, and I thought, I got to meet this guy. And so that's how it all started for me. I talked to him about it. He had roughly 30 different disease programs he was working on. Basically had too much to do. And so more or less my first foray into Angelman syndrome was a gift from Dr. Gray to work on this idea he had for permanently activating the paternal allele or UB3A. So that's not so inspirational, but what I would say is that while that is not so great to hear, I've been able to stay in it because prior to working with gene therapy, I never really had any exposure to the individuals I would theoretically be helping. And so that's where I think the inspiration comes from with my stories, coming to these conferences here, meeting you all, meeting the patients, meeting everybody, the families, and knowing what we're working for. Thank you.
Right. So I do have a few questions that I've guessed we might want to ask the panel. So I was just going to start with, I can barely see them. Oh, I can see them now. So as I mentioned earlier, ASOs, the antisense oligonucleotides, are now in the clinic. So I want to have the panel maybe talk about the other treatment approaches that are currently being explored. So go ahead.
Hello, can you hear me? Okay, good.
They'll turn it up and down. Is what's going
on. All right. Is this one on? All right, good. So you all have heard a lot about ASOs, antisense oligonucleotides, and maybe some of you have your children enrolled in these clinical trials. And as you know, these are relatively invasive injections. You have to come back to the hospital every 3 to 4 months to receive an injection. And so what our lab is trying to pioneer is, and our lab and many others actually, is a different approach where you could essentially get a single injection that would last a lifetime. And so ASOs work by targeting the UB3A antisense transcript and stopping this thing, turning it down. You might have heard about these other technologies known as genome editors or CRISPR-Cas. A Nobel Prize was awarded for this major discovery several years ago. These are, fun. They're really revolutionary new technologies that allow you to go into the genome and very precisely find a specific site and modify it in some way. The genome is made up of billions of letters. You can turn those letters into other letters, and those letters can include things that stop transcripts or stop the antisense transcript. And so we're working with these various genome editors to go in and permanently modify the genome so that the antisense transcript is no longer expressed, and allowing for permanent re-expression of the paternal copy of UB3A. And so obviously this would be exciting. That this would be an alternative to the ASOs, but these new therapies will also carry a higher level of risk. ASOs, when you inject them, they don't last forever, and so that's a good thing, and it's also a bad thing. In the context of a new drug, if there's side effects, the ASOs you can just wash out and the effect will go away. But with a genome editor, the effect is permanent. You can't go back and fix the changes that took place. And so there's just a higher bar for safety. And so people are exploring many different ways of using these genome editors to target the antisense transcript. And then another thing that we've been really focused on is how do you assess the safety and efficacy of something that is permanent before you take it into a human that has Angelman syndrome. And so we're exploring other ways of testing human therapeutics in animal models so we can get at efficacy, both safety and efficacy issues in our research.
So, I mean, they've already explained this quite thoroughly. I will just add that there are some other things that we are exploring at the moment are novel viral capsids. And so the benchmark right now for gene therapy, at least single dose gene therapy with viral vectors, is AAV9. That's been around for, I don't know, probably close to a decade or more, it's fantastic. But the distribution in the brain is limited. And so at best, you'll get treatment, but not a cure. And so one of the things that are currently being explored are the developments of these new viral capsids, some of which have had tremendous biodistribution within the brain, several-fold better than AAV9. And so that means that pretty much all of these approaches could, in theory, work now in humans in a way that wasn't available before.
Yeah, I have nothing to add.
So I guess since Ben is not here, I'm going to put on my Ben Philpott cap, since I do talk to him all the time, and say some of the things he might have said. So his lab is using other approaches to advance alternatives to ASOs, one of which is a gene replacement. And so, as you know, UB3A is missing in individuals with Angelman syndrome. And so another way of fixing the disorder, so to speak, is to bring back or add back a functional copy of UB3A. And so using these adeno-associated viruses that Ryan just talked about, you can add a functional copy of UB3A into a vector and then inject that into the brain. The trick with this is that there are different versions of UB3A that Ipe's lab has really done a nice job of describing. And in the brain, they're not all just expressed at random levels. There's a certain ratio of the different versions. And so what Ben's lab attempted to do, and I would say it was quite successful, is to mimic the ratio of the different versions in this single gene therapy vector so that when it's added back, the brain is getting not only the right levels of UB3A, but the right versions of UB3A. And he's published a really nice paper showing that this gene replacement therapy is very efficacious, very effective in the animal model of Angelman syndrome. And I know his lab is continuing to pursue that, I believe with drug companies. So there's the hope that this could be something that moves forward even more rapidly. And you also heard from the last talk, for those of you who were here, about a small molecule that his lab was developing. I had mentioned this topoisomerase inhibitor that we had discovered with Ben's lab years ago, but it had side effects. And so he continued to do more screening. He's like very persistent. Just like whenever he got his hands on new drug libraries, he would test them. And so this has been going on for a long time. And then he just reported in a paper and at this meeting a new small molecule that is not a topoisomerase inhibitor that seems to have a better safety profile, at least in animals, to turn on UBE3A. And what is really pretty amazing is that this can be delivered orally. So an oral, single oral administration turns on paternal UBE3A within a day in mice. And so there's still a lot that needs to be done because all of this is just being currently in animal models. But this could provide another treatment approach where you just would take a pill to turn on UBE3A.
So my next follow-up question, which I think we may have already kind of talked about a little bit, is about, well, let's talk about this some more. So what differentiates these approaches? What are the pros and cons? So we've already touched on, I'm going to try to set this up for Ipe, but we've already talked about the pros and cons a little bit. ASOs, apparently very effective from everything we've seen in all the models, and now in the clinic, we've got some evidence of efficacy. But requires repeat injections through a lumbar puncture every 4 to 6 months, maybe is where we're headed. The gene therapies would hopefully be one and done, but the very effective treatments, but it's a single in treatment that would be there for a lifetime, and we have concerns about safety. Whereas the small molecules are the advantage there is orally active, so we don't have to deal with the injection. So Ryan has taken the microphone. Maybe he's ready to talk a little bit more about the pros and cons of these different approaches. And although maybe he's going to hand it off to Ipe.
Yeah, so that has been discussed quite a bit here, but ASOs, they have evolved quite a bit in the past several years. So there are modifications now to where it translocates directly to the nucleus, widespread distribution throughout the brain, but the half-life is quite short. And so in a single year, the patient would need 3 or 4 injections into the spinal cord, into the spinal fluid, excuse me, every year. But the efficacy has been tremendous in the mouse models. And from what I've heard, at least with the clinical trials there, it's working tremendously as well. I'll just speak to my approach for now, which has some relevance to some of the other gene therapy approaches. So I'm working on an shRNA approach, which basically is similar to the ASO. It's an RNA interference. It's using the viral DNA, which does not go away, as best as we know, to non-dividing cells. It should stay there as long as the neurons or the cells are alive and healthy, or at least working. Some downfalls to this, non-specific binding. It's something we can control for as much as possible, but we don't know until we do diligent safety tests on it to make sure that, yes, it's most likely binding where we want to, but where else is it binding that we don't want to? And we need to know that before we go into humans with that therapy. Another drawback is that it can compete with endogenous molecules called microRNA within the cell, which can lead to toxicity in certain cell types. That's been demonstrated in rodents, not in humans. And so we are trying to mitigate those concerns with our diligent safety tests. RNA-seq, basically, we inject the organism, the model, and then we get the cells and we look at the entire transcriptome, all of the RNA, and make sure that only what we want changed has been changed. And if there's anything else that's been changed, it doesn't matter. That's the goal. That's what we want. That's what I have to say about that.
I just want to add one thing, because we are all focused very much on our genetic technologies, and bringing back the UBE3A gene is the ultimate dream of every clinician and scientist and parent, maybe, because that's really what's wrong. that we should not forget there's still an interest in molecules that are not really what we call disease modifying in a way that you actually bring back the UBT gene, but can really help just to alleviate some of the symptoms. And still a lot of children have epilepsy, and I still think we can do much better on that by having good anti-epileptic drugs. And this is still something that is going on. I mean, we test in our lab still small molecules that we don't expect is going to entirely cure the animals, but really may actually be very effective in the epilepsy, show some impairments in their anxiety behavior, etc. And these are still ongoing efforts, and I think we should keep on doing that until we have the magic bullet.
As everyone was talking, I was thinking of another pro/con. So all of these technologies, like when you think about them, it's kind of like magic. The things that we can do to the genome in a specific way, is very new. We haven't been able to do this before. But one of the things that we often don't talk about, we just say, yeah, we can modify the genome, and you just assume that this is going to work in the brain. But the reality is that the brain is this like specialized organ. There's this thing called the blood-brain barrier that makes it very difficult to get things into the brain. And this is something that this is the reason why the ASOs need to be injected into the spinal fluid. You can't take an oral ASO because it would get destroyed by the stomach enzymes. It just wouldn't get into the brain. So you have to pretty much deliver these things into the CSF to access the brain. ASOs do, as you know, transport well into the CNS, into the brain. But a lot of these other technologies that we've been talking about, like the CRISPR-based approaches and even these shRNA approaches, are relying on a vector called AAV, and it's known that that these don't work that well to deliver throughout the brain of adult animals. So there's a real need for more research at every level to find better delivery approaches to get things into the brain. So, in many respects, the brain is like the final frontier for medicine. Because of this blood-brain barrier, it can be a challenge to get things into the brain. These genome editor technologies and other approaches are truly revolutionary, but we need to couple these with new ways of unlocking the blood-brain barrier and getting them in so that we can modify the genome of the neurons. And so this is a major problem for the field that I think lots of labs, lots of companies are working on. So there's a lot of hope, but you have to stay tuned in many cases.
I'm gonna pause and just remind people you don't need to raise your hand. I'm looking around the audience and I see hands being raised, and I'm realizing that some of them are not necessarily parents; they're individuals with Angelman syndrome raising their hand. So if you do have a question, and even if the Angelman individuals want to ask a question, come up to the mics, and we'll start taking questions from the audience whenever you come up. I'm going to go on to the next pre-planned question, if you will. And I wanted to talk a little bit more about the safety side of things. So what is, we're talking about things that are being evaluated in animal models, in vitro models, cellular models and things. Can you speak to what is being done to ensure safety?
Thank you. So to ensure safety, at least I'll speak for gene therapy right now, for what we would do for our approach if we actually get to that stage. So first we verify, we show efficacy at first, and then we move on to non-human primate safety studies, and that would include neurocognitive testing, behavior testing, toxicity. Afterwards, we would take tissue samples from throughout the organism's body, and we would test it for, like I mentioned before, for any signs of toxicity, any signs of off-target effects, things like that. And also while we're doing our efficacy studies, we are monitoring for unexpected phenomena that occur in our models, early death or anything like that. We're monitoring that. We have to record it. If we come to the FDA, they have to know what happened to each mouse and why, as best we can explain it.
Yeah, of course, besides the mice, many of the drugs are also tested now in non-human primates. So as a next step.
I'll add yes, thank you. The FDA keeps a close eye on this, and generally at least 2 animal models are required typically And so they do evaluate all the data, and everything needs to go through the FDA before it can go into the clinic as well. So they have a very, very important key part of this whole process. For these approaches, and again, I'll just go on to the next question. Would you like to ask a question here? We can. Right, we're going to jump in with a question from the audience, please. Just to clarify the question, you're seeing in the individuals with Angelman syndrome that you're seeing here? Yes. Right. Right.
Yeah, so within the deletion, we have to first realize that there are also different genetic subtypes. So some of the kids have a larger deletion than other kids. So that may also means that there are different genes are going to be deleted. But besides that, it's also really important to realize that when we use our mouse studies, we have a highly so-called defined genetic background, and that is basically that almost all mice have the same genetic background. But we know that the genetic background makes a big difference because, for instance, one of our favorite tests is a test where we induce a seizure by a loud noise. Well, when we have the black mouse with UBTA, this mouse will not show any seizure, but then we have a mouse that has a skin called agouti, like grayish mouse, immediately will show a seizure when you do an audiogenic seizure. So they have the exact same mutation, Angelman syndrome mutation, yet the outcome is really determined by the other genes in the genome that can, to some extent, compensate more or less for the fact that UBTA is not there. And I think this is probably also why we see a lot of variation among the children. Earlier this morning, actually, I was just talking to a researcher and we looked at also, doesn't matter whether if you had your first seizure at age 1, how does that change the outcome as opposed to having no seizure or having a very late seizure during development? And also there, I mean, this also from other disorders, it really matters when you get those first seizures, because if the seizures are early and they are poorly controlled, that also will really affect the outcome. So again, adding variation to something that is not necessarily a genetic variation. So multiple causes really that cause the variation between individuals. I was anticipating you. Yeah, so because for the last 20 years, we only had a UBTA mouse model, all our focus was on the UBTA mouse model. And also, of course, the reason that we all focused on that is that because we know that no matter what mutation you have with respect to Angelman syndrome, they all have in common that there is no UBTA gene. So we looked in our mouse models, what happens if you bring UBTA back in the UBTA mouse model. But more recently, we also took the mouse that has the deletion and then asked ourselves the question, what happens now if you lack 10 other genes, also including UBTA, and then you add back UBTA? Does it matter that the other 10 genes are still deleted? And so far in our mice, but we still have to do further studies, but all the tests that I showed earlier this morning that are affected in the mouse model of Angelman syndrome, all those tests are completely rescued if you bring back the UBTA gene, despite the fact that you have 10 genes that you did not actually rescue and left untouched. So that really suggests that indeed UBTA is really, really the main driver, also in the terms of for the deletion patients, and that if you were to get UBTA back, maybe to some extent those 10 genes would matter, but not to the mice suggest it's not to a very large extent that they actually contribute to the phenotype.
Tim, did you wander up with a question? Did you?
Yeah, I guess, we definitely we're not experts when it comes to clinical trials. So it'd be hard to know how AI could benefit in that regard. Yeah, our
Just human research in general, could AI kind of coalesce something fast enough to data?
Okay, I'm gonna put my science hat on. Sorry, my mic's on. I'll put my science. Sorry. Is my mic, there we go. I'm gonna put on my scientific hat and try to just follow up on Tim. So, I coincidentally, I had lunch with a colleague of an old friend of mine just last week who's been involved in drug discovery research for 30 years. And he's on a break between jobs and he said he's doing a deep dive into AI. So I've been thinking about this in AI. The important thing to recognize from my perspective is that computational bioinformatics, computational science has been used in drug discovery for decades. Okay. And so a lot of what we're talking about now in connection with, which is now being called artificial intelligence and AI, has been developed and is in practice and is very aggressively used to support everything that we do. So example, with respect to the things these guys might do, they generate large amounts of data. And we now can go through genomes and generate all this genomic data and analyze it computationally and get all that filtered. So that's basically things that are already going on and that we've been doing for many, many years, where we're using computers and computational approaches to drive the research forward. The new stuff you're hearing about has a lot of potential. And I was just talking in the last couple of days with people about this. Ways we might be, where are there things missing and where can we use it? The important thing is that we need good data to go in. And that's, if you think about ChatGPT and these other things, it's only as good, it'll generate information or answers to your questions, but they're only as good as the data that goes in. So the very first thing that we need to do is have great data going in so that when you analyze it, you can believe and have reliable results. So for example, what we're doing separate from this, the lateral learning network, we're consolidating all that data. That's the sort of the first step where we need all of that data organized and in great shape so that we can then apply these computational approaches to analyzing and dealing with it in more detail. So a little off topic for this, but very much it's something that we're looking at for the future.
So maybe I can also add just like one other thing. So we do use increasingly artificial intelligence in the lab for basic research. So when we're studying mice, there are standard ways of looking at behavioral deficits in the animal models, but they're pretty crude, basic measures. With AI, people have developed more sophisticated ways of characterizing alterations in behavior in animal models. So you can take videos of animals and find subtle differences. And so that's the work that our lab and many other labs are doing. And in one of the talks yesterday, you heard about how verbal and nonverbal communication is an issue in Angelman. It turns out that Angelman model mice don't vocalize normally, and it's very hard for a human to understand or quantify those deficits in the mouse. But with artificial intelligence, you can actually rigorously quantify the vocalization deficits in the animals so that you can assess how well does the drug work in the animal model to bring back some of the vocalization deficits that we're seeing.
I think we have another question here.
Thank you guys. So you were talking about putting the UBA 3A in when it's gone. Now, would there be a certain time period that you guys could do that? Like, do you know when that gets deleted? And if you do you know like what time period you would want to do it? Like if they were still in the womb, for instance, if you would do that right then and there, or if you would wait till baby fully develops and was out of the womb.
Yes, so this question is a very important question. When do we treat? Because it's also really important how you design your trial, because if you're going to take a population and then turns out that the population was too old, then maybe you throw away a beautiful drug because you designed the trial wrongly. So already like 5-6 years ago, we answered exactly that question in mice and said, okay, what happens if we just by a drug switch on the UBT gene at different ages? And we looked at an early embryonic phase. Then all the mice got 100% cured. And then we looked at what happens if the mice are just born and what happens if you do it in adolescence. And also really what happens if you have a mouse that is by far all criteria adult and then switch on back the gene. And yeah, I talked about it a little bit this morning, but basically what we see is that it really also depends on what it is that you're looking for. So for instance, the ability for synapses to change, synaptic plasticity, that can be changed no matter how old the animals were. But there were certain like behaviors that probably the mice already born, they show already their behavior very early on, that we could not change if we injected if we put on the UBT gene too late. So then those mice just stayed in that state. So it really was, there are what we call critical periods where you can inject a gene and then you can rescue certain phenotypes. But it really depends on what we call the phenotype that you're talking about that determines what the critical period is. So there's not just one magic word that says this is it, and beyond that there is nothing. Probably like if you inject an adolescent individual with Angelman syndrome, some of those things might not entirely be reversed or not reversed at all, whereas other things can still very much improve. Overall, of course, it's brain development, so early is always better. Indeed, we think that we would like to that ideally you can start these approaches as soon as you have a genetic diagnosis, or maybe if you have a good prenatal screening program already much earlier. And there, Mark can say something about that.
Yeah, and I think, yeah, obviously it's important to explore treatment options across the lifespan. But one of the goals of our work and others is to see how young you can go. So theoretically, Angelman can be diagnosed at conception, and so there are ways of. I mean, if one were to do in vitro fertilization, for example, you could pick up the deletion at that age, but that's usually not the way most people have children. So there are genetic tests that can be used during pregnancy. And they have the ability to detect the deletion that causes Angelman. They're not widely used, but there are these tests that are increasingly being utilized called NIPT or non-invasive prenatal tests. So you can take an additional 5 milliliters of blood from a mom as early as 10 weeks post-conception, and there is fetal DNA that's circulating in the mom's blood. And the sequencing technologies are so powerful now, even though the amount of DNA from the fetus is small, you can detect that and identify changes. This is really good at picking up the sex of the fetus, Down syndrome, other large chromosomal abnormalities, but it can also detect the deletion that causes Angelman syndrome. These are not diagnostics; they're screening tools. And so, if this test shows the mom has a deletion, a follow up with an invasive test like an amniocentesis or a chorionic villus sampling would be needed. But then, so the technologies to diagnose Angelman prenatally exist. So we don't need new technologies in that area. But we get back to this safety, the risk benefit. No one has ever injected any kind of gene therapy prenatally for any disorder, and so that's I often talk about this as the prenatal barrier. If you have a baby that's immediately born, you could theoretically treat that child, and you're really only focusing the therapy on the child. But if it was the day before, you have to consider the mom as well, and the risk benefit not only to the fetus but also to the mom. And so the need for demonstrating safety and efficacy when you go into this prenatal period is going to be much, much higher than postnatally. And so these are things that many groups are talking about for a variety of disorders. But at this point, this is not, no therapies, genome therapies have been applied at the prenatal stage.
Guys.
I just wanted to add one thing about you're touching on really the future of pediatric medicine right there. That if you want to look around the bend, we're talking about genetic diagnosis in the womb and a treatment being available off the shelf to give in the womb. That would be ideal. There's probably all of these approaches that we've been talking about would be benefited if it was given earlier. That's probably without question. And so you're absolutely right to ask that question. Thank you.
So I've been getting the 5-minute sign from Amanda, so I'm going to go to my last question, which I think is a good way to wrap it up for the panel here. What can patient advocacy groups do to support these efforts, and what can families do to support these efforts?
I guess. Hello? Yeah. And one of the big things is just advocacy for more research, especially at the federal level. The NIH is this behemoth, has a huge budget, and I think everyone maybe just assumes that the NIH is putting money into Angelman syndrome or whatever disorder or disease somebody's child may be afflicted with. But the reality is, even though the budget is huge, the money is limited. And so they have to make decisions about where they put their ultimately limited resources. And so the more the government, Congress, the administrators at the NIH hear from parents about the importance of Angelman syndrome research, they'll be more likely to direct more of their limited funding towards this disorder versus other disorders. And obviously that would benefit the community. And I feel like of course I'm biased, but I think Angelman is the most important disorder to move forward with a variety of these genetic therapies, because there is a real clear path to turning on the paternal copy of UB3A using a variety of methods. And like in many of these cases, it's basic research that really needs to be studied in animal models and other models to demonstrate how well these things work, how well can we get these into the brain of young animals as well as adult animals, and that's going to take a lot of research, and that can't be done without substantial amounts of funding. This is not cheap.
Yeah, I echo all of that. I want to say two things. I mean, like the money that the ASF provided has been instrumental to all the work that we do in our lab. For instance, all the mice models were actually paid by the ASF to generate them. And once you have such a mouse model, then you can actually ask for the bigger grants. I mean, I live in the Netherlands, so we don't have the NIH money, but we can ask for bigger grants because we already de-risked the grant application by already showing that we have the mice and that the mice are viable. So this seed money that the ASF has provided all of us, I think, has been instrumental. And if this question were to be asked when I was in the Netherlands, I said, you have to really say that animal work is absolutely necessary to make progress, because like a few months ago, the University of Utrecht, one of the biggest universities, decided to close down all animal facilities. And now even at the Erasmus MC, they're talking about closing down the animal facilities because there's so much objection to work with animals. And in particular, people that are healthy, think that we could do without animal research. But if you have an affected child and you want to study the questions that have been asked today, like when do we need to inject, etc., animal research is unfortunately still very important. So if you find people that are really against animal research, tell them that it's still important to get drugs.
So it's hard to say what advocacy groups can be doing more than what you guys are doing already. I will just say that until there is a cure for Angelman syndrome, there's not a cure for Angelman syndrome. And so it's finding the balance between, at least for the research, finding a balance between funding one project and going with it all the way and funding new things that could possibly be even better. And so that's a tough balance to find. And so that's just something that advocacy groups need to do. For families, I mean, you guys are an inspiration to us. Our job requires a lot of perseverance, and nobody knows perseverance better than you all. So thank you all for that. If you ever find that you're cynical about the research, about the science, I mean, you guys also have perspective, but some perspective from our side is that for pretty much all of medical history, the brain has been an impregnable fortress. Really, the only way to do anything with the brain disorder would be to go in there and remove the diseased neurons and whatever healthy tissue was deemed acceptable. So it wasn't trying to fix the neurons or help neurons. That was it. And then we're looking at something so unimaginably small. You may look at your children and think that there's something big going on, but it's so unimaginably small. A B-2 bomber, I love telling this story, by the way. A B-2 bomber has a wingspan of roughly 2 football fields. Imagine it is 1,000 times bigger than that, and a single bolt came loose, and it can no longer fly. So for the longest time, where researchers started, we were just trying to find where to go, what to do. And that if you think that that's hard, while the plane is flying, all of a sudden the bolt comes loose and we're trying to fix it. That's where we started. And compared to where we are now, the progress has been tremendous. And so just keep faith that something's going to come up, and we will too.
So I'm gonna end it all there, but I want to thank all of you for being here and for supporting what we're doing. And if you have additional questions, I'll be here all week. I'm not sure how long my panel will be here. I'll be here all week, so please find me in the pool, in our meeting rooms, or I'll be available. And also Becky Burdine, who many of you heard talk earlier, she's doing the genetics talks right now, but she's available too. So we'll be here all week. Thank you very much for your support, and have a great time here at the conference.
Hey, let's see. Okay, thank you guys so much. Once again, thank you to the researchers. We're going to jump right back into the next session, because we're trying to get back on schedule. So if I could go ahead and have my amazing next panelist come on up, and I'll introduce you guys as you come up. So this next session is 3 years into Angelman clinical trials and what we know, and we have an amazing group of panelists here who have been working in the Angelman space for probably collectively 70, 80 years at this point. It probably feels that way. But I would love to invite up to the stage first Dr. Lynn Bird. Come on up. From Rady Children's Hospital, we have Dr. Elizabeth Berry Kravis from the Rush University Medical Center, Dr. Wen-Han Tan from Boston Children, and Anjali Sadween from Boston Children as well. So thank you guys so much for being here. I've been told that I'm going to pass it on to you guys, and you know what you're doing. Correct? Awesome. If not, I'm in trouble. And so I'm going to jump right in, because I know that you guys will probably have a lot of questions as well. So after they're done with their presentation, just a reminder, there's microphones up here if you would like to come up and ask a question, but we will go ahead and pass it on and get started. Thank you guys so much for being here.
Yes, I'm good. As long as the slides advance, I'm good. All right. This is just all of us who you all know. And so the session aims here are to provide an overview of the clinical trials process, to describe experience to date, as well as a little bit of a discussion of where we're going from here, and to review considerations about for how you and us can get the most out of your trial participation for the greater good of everyone. So this is the agenda. I'm going to talk a little bit about the clinical trials process in general. And then Lynn is going to talk about the ASO mechanism and the current ASO trials, because that's mainly what we've had so far. And Angela is going to talk about outcome measures. And then Wenhan and I are going to talk about some best practices for trial participation going forward. And then we're hopefully going to have enough time for you all to ask questions. Okay, so this is just a picture of clinical trial development, which shows you how long and complex it is. We have to get an idea for how we're going to treat a disease, and then we have to verify that we're targeting the disease. We have to develop assays to prove that. We have to find compounds and screen them, and then find which ones are the best ones. And then we have to have preclinical efficacy in animal models, safety and toxicity data. And then finally, after all that work, we get to actually give the drug to a person in clinic. And that's when we get phase 1 and phase 2 trials followed by phase 3 trials. So on average, it takes 13 years to develop a drug with 5 years in clinical trials. And you're going to, on average, start with 9 drugs to get one approved. And this costs, as you can imagine, gazillions of dollars. So the Angelman ASO trial started in 2020. So you might think, well, 5 years, 2025, we should be done. But some of these trials some of the development process can be longer if the trials have to be long trials, like you can't see the effect of the drug in 2 weeks and you have to look at it for as long of a time as 6 months or a year. And how long it takes in the trials depends on the rate of recruitment, which depends on whether we have enough participants that want to be in the trial, site resources, how fast can the sites enroll participants. Personally, I would like to believe that an ASO will be approved by 2027. I have a video about that if anyone wants to see it. And that's the hope. It might not happen quite that fast, but that's what we hope. Okay, so once we do actually get into clinical trials, this is the process. You're going to have this is the standard drug development process that I'm showing you right here. There's going to be a phase 1 study, which is usually normal volunteers. And so those normal people they give the drug to these people just to make sure that no clinical disaster happens. And they kind of dose one person at a time. And then after you prove that it's safe in a limited number of normal volunteers, you go to phase 2, which is a safety study where you're studying more patients, usually around sort of in the range of 30 to 100 patients. And then you look at safety, but you also get an idea of whether there's a signal for effectiveness or efficacy. And then you do a phase 3 trial, which proves whether the drug is effective. When patients finish the phase 3 trial, often they stay in a long-term extension to be exposed to the drug for over a year so that there can be long-term safety data that's presented to the FDA when the drug gets approved. And then after FDA approval, in some cases, you run a phase 4 or post-marketing trial to look at some specific safety issue or to try to find out what happens long-term with the drug. But in rare disease, sometimes this process is a little bit different. And phase 1 and 2 may be combined into one study, like we've seen with the ASO trials for Angelman syndrome. And especially if we're giving an ASO or a gene therapy, we're giving some kind of compound that's specific to the disease and has to be delivered like intrathecally, and there's more safety risk, then usually you want to do that in the disease population because it's too risky for normal volunteers just to be exposed to that kind of a drug. And then the phase 1/2 trial may be associated with a long-term extension because the disease may be a disease where the treatment could evolve over a long time, and you really want to collect information on what the drug is doing over a long period of time. And then the phase 3 patients may go into the same long-term extension. And this is kind of what the development process for the ASOs in Angelman syndrome is looking like right now. And then you have the same issue after FDA approval, but especially in a developmental disorder where change might take a very long time to see, especially in cognitive disorders. Sometimes phase 4 post-marketing trials are really important to actually find out what happens to the patients long-term after they go on a treatment. Okay, so that is the process of how we have to go through to develop these medications. And I'm going to turn it over. Oh, wait, I have like where we are in the process. So GTX 102, the Ultragenyx drug, is just entering a long-term extension, and we'll be starting phase 3 soon. The Ionis ASO is in the same place. Rugonersen is at the end of phase 1/2, but will go forward only if Roche can find a partner. And then Allogabat and NNZ-2591, I just want to mention, these are molecules that work on downstream signaling in neural cells, and they're just oral medications, and they're in the middle of being looked at in phase 1 to 2 to determine if they look like they have enough signal to go forward. Okay, and so now I'm going to turn it over to Lynn, who's going to talk to you about the ASO mechanism and trials in Angelman.
Hi, good morning. This feels a little redundant since we've been talking about ASOs all morning, but just for those who may not have caught what the mechanism is like, I'll remind you that we all get 2 copies of our chromosome 15, one that we inherit from mom and one that we inherit from dad. And the gene of interest is UBE3A, and that gene is only expressed from mom's chromosome. Dad's chromosome is asleep. So a gene is a long piece of DNA that then gets transcribed into RNA, which is the messenger, and the messenger then gets translated into protein. So we need to have the gene make the RNA and ultimately make the protein. So that's what happens on the maternal gene. On the paternal gene, however, there is a block in transcription. So we don't get RNA made, and therefore we don't get protein made. And so obviously, if you have a problem with the way your maternal gene is performing, your paternal gene is not able to help out because it's repressed. Now, it's important to note that every type of Angelman syndrome has at least one sleeping or silenced copy of UBE3A, so at least one paternally imprinted copy of UBE3A that we could potentially activate or wake up. So how is that repression? How is that repression accomplished? Well, there's this piece of RNA that comes from the opposite direction of the direction that the gene would normally be read in, and that's called the antisense transcript. And that long piece of RNA interferes with the gene being able to be transcribed in the correct direction. And so it's effectively silencing the gene, repressing the gene, or has put it to sleep. So what we want to do with ASOs is that we want to try to interfere with that antisense transcript. So on the left at the top, I've got a piece of RNA that's the antisense transcript. And what we want to do is attach something that will destroy it. And that something is called an ASO, an antisense oligonucleotide. This is basically a piece of artificial DNA that is engineered to exactly match a section of the antisense transcript. It matches perfectly. So there's only one place in our entire genome that this antisense oligonucleotide can match and bind to. And once that ASO binds to the antisense transcript, it triggers a mechanism that we all have inside every cell in our body, which is the RNase H mechanism. And RNase H looks and says, oh, wait a minute, RNA and DNA are not supposed to get together. This is not supposed to happen. So the RNase H chops apart the RNA. So we are using an artificial piece of DNA to tell the body to chop apart this antisense transcript, which then we hope will release the paternal gene to be able to make RNA and then ultimately protein. So on the right side, you can see that in the untreated situation, you have your antisense transcript in there. It's repressing the paternal gene. The mother's gene is either absent or has a mistake in it, and so it can't work. In the treated section, bottom right, now you give an ASO, you get rid of that antisense transcript, and now you've got the gene that can be expressed. So we have, as Dr. Barry Kravis mentioned, 3 trials going on. They're in various stages of phase 1, 2, rolling over into long-term extensions so that we can get long-term safety data. And with that, I think we'll go on to Anjali talking about the outcome measures that we're using in these trials.
Hi, I'm Anjali. I'm going to be talking about the outcome measures that we use in these trials. I know as families come into trials, a lot is expected of them. They have to fill out tons of questionnaires. Your child has to go through a lot of testing. So I just want to talk a little bit about some of the outcome measures. I know there's a whole battery of questionnaires, tests, or video assessments that you do. So I'm just going to talk about the key ones that I used in these clinical trials as outcome measures. So when we talk about outcome measures, we talk about 4 categories. We talk about a performance-based outcome measure. A performance-based outcome measure is when we have an individual with AS come and do something. So we'll test them. We'll see how they walk. We'll see how they respond to things we give them. We have a patient-reported outcome measure. Patient-reported is when the patient itself says something. Because our individuals with AS are nonverbal, in most of these trials, they're not using any patient-reported outcome measures. You then have a observer-reported outcome measure where you as a caregiver will fill out what you see about your child, what you know about your child. And then we have clinician-reported outcome measures where the clinician will ask you a lot of questions, but then they'll make a decision of what they are seeing and where they find your child. And it's really important that all these outcome measures are FDA approved and so that it can be used in trials. So what domains of development are assessed in a clinical trial? A lot of domains are assessed. We're looking at cognition. We're looking how your child is thinking. What's their memory like? What's their problem solving like? We ask you questions about their adaptive functioning. We're asking you questions about like, how is he at home? What's toileting like? What's feeding like? How does he do socially? We're interested in their receptive language skills. What does your child understand? What kind of instructions does he follow? Expressively, we're interested in what does your child vocalize? How does he use AAC? How does he use manual signs to communicate his needs? When we're looking expressively, we're looking at any way of communicating. How does your child communicate? We're looking at fine motor skills. How is your child grasping things, navigating their environment, turning doors? We're looking at gross motor skills. How is your child navigating the environment? Is your child walking yet? How do they do on stairs? So these are the different domains that are assessed in a clinical trial. One of the most critical outcome measures that you probably see and are used in all the INS, in Ultragenyx, and the Tangelo trial was the Bayley scales of infant development. The Bayley scale of infant development has been used because it assesses multiple domains of development. It assesses cognition, it assesses receptive and expressive communication, it assesses fine and gross motor skills. This is the performance-based assessment. This is the one test where your child would come into the clinic and you'll have a psychologist or a trained clinician do the test with them. The Bayley, even though it's used for younger children, it's been normed for number of children up to 42 months. It's used for all individuals with Angelman syndrome, and it's been used in many studies. And so we have a comparison data of like, how do the children look on the Bayley without being on the trial, and how do they look on the trial itself? And it's very sensitive to change. The couple of things that we want to make sure that when you're using the Bayley in the trial, it's important to administer it at a time when your child is most likely to cooperate. I usually believe the morning time is the best. You also want to make sure your child's had a good night of sleep, right? You want to make sure this is not done after a blood draw. This is not done after an EEG when your child is in distress. You want to make sure it's scheduled at a time where your child is available to be fully present to do the test. You want to make sure that the Bailey is as much as possible done by the same clinician in a trial. So the one who's on the first visit sees them on the second visit and the third visit. And how can parents help? I think you know your child best. You know, some of these items are not motivating for children. So if they're treats, they're snacks that you know your child will work for, bring them to the session so that the psychologist and clinician can use it to get your child to be motivated to do it. So that's the Bailey scales. The next test that I'm going to be talking about is the Vineland Adaptive Behavior Scales. This is a parent-completed questionnaire, so it's an observer-reported. So this is where a clinician will ask you questions about how your child is doing. We're looking at how your child is doing at home in communication, in activities of daily living. How is your child socially? How does he navigate his environment? The clinician is going to spend some time with you, asking you questions and then making ratings about it. It's really important that sufficient time is kept aside to do this interview. We usually allot about an hour and a half to do this. We want to make sure that this is done at a time where the parent is fully present. So if you're doing it in clinic, you make sure your child is done. We make sure there's somebody else taking care of your child so you can fully attend to this. If we are trying to do this remotely or virtually, we schedule it at a time when the child is in school. You know, the parent has some free time so they can be fully present to do this interview with the clinician. We want to make sure the same parent completes it because mom and dad always have different responses. So you want to make sure if mom does it the first time, she does it the second time, and she does it the third time. So it's really, really important that it's the same parent doing it. And as same thing, it's important that the same clinician does it, right? Because ratings change. We want to make sure it's the same clinician doing it the first time and the second time and the third time. The last measure that I'm going to be talking about is a parent-completed questionnaire. This is the ORCA, which is the Observer Reported Communication Ability Scale. It was specifically designed for individuals with AS. It assesses communication at different domains, expressive communication, how your child communicates, what your child understands, as well as how does your child communicate socially. It accommodates multiple communication modalities. It's filled by the caregiver, and it takes about 15 to 20 minutes to complete. Some of the things that you want to keep in mind in looking at the ORCA in a clinical trial is make sure that there's sufficient time kept aside to do it, and make sure there's the same caregiver completes it every time. Those are the important considerations that you need to keep in for clinical trials. I'm going to have Winhan who's going to talk about the next outcome measures.
All right, so thank you, Dr. Salwani. So you've heard a lot about developmental outcome measures, right? So the Bailey, the Vineland, the ORCA, as sort of assessing different components of development, which obviously is absolutely critical. But there's a lot more to Angelman syndrome than development. There's sleep, there are seizures, and there are other components. So we now have two clinician-administered outcome measures, and they are called clinical global impression scales. The emphasis is really on global and it's an impression, right? So this is looking not at very specific aspects of any domain, but really your child as a whole. So there are two components to the CGI. There is severity, i.e. how's your child been doing in the last week or 2 weeks, depending on the scale that's used, for in the different components that you look at, where we'll go through in a minute. And how has that changed? So in the context of a clinical trial, what we want to know is, compared to baseline, has there been a change, which may be better or worse? Right? So if the drug is making certain components of Angelman syndrome worse, we need to know about that. So one of the two CGI scales that we're currently using is the one that was developed initially by GeneTX and then by Ultragenyx. And it assesses the different components that you can see. So sleep, behaviors, communication, gross motor, fine motor skills, and activities of daily living. Now, in this ultragenic scale, they have, at least in the severity scale, they have what we call anchors. So the clinician will ask you about sleep. How many hours of sleep do you get a night? Has the child been getting a night? How long has it taken a child to fall asleep? Does the child wake up in the middle of the night, etc.? So what the clinician would do is to take all the information into consideration and then assign a number, a score to each of these domains, right? So there are specific questions that you'll be asked, and based on your responses to those questions, we will get an overall impression of what your child is like. I will say that the way the scale is designed is that the middle of the road is basically a typical score for a child with Angelman syndrome based on that age. So it is dependent on age as well. Now, the other scale is the Roche CGI, so developed by Roche. They have very similar domains as the Ultragenics. In fact, most of the same domains, but they include seizures and overall cognition, which the Ultragenic scale does not specifically assess. However, the Roche scale does not have specific anchors. So the way we administer it is I would ask you something like, well, how is Johnny's sleep? Has he been waking up at night? So there are some sort of non-specific questions that we will ask you, and based on your responses, we will try to assess whether your child's sleep, for example, or behaviors, etc., are typical or average for that, for a child of that age. So switching gears a little bit. So those are all sort of outcome measures. Now, in any clinical trial, safety is really important. And yes, we want to find a drug that works, absolutely. But we also need to find a drug that is safe, that will not cause side effects or undesirable side effects. So it's really important to think about safety. And this is why it's really important that you work with the doctors, the clinicians that are conducting the clinical trials in your child. It's really important that you tell us about all your current and recent illnesses. Because if your child has just had the cold or COVID, touch wood, or anything like that, that can impact the risk of sedation or anesthesia. We don't want to put any one of your children at risk unnecessarily. So if they have had a snuffer, they have had a cough, please do let us know. It does not mean that we will delay or cancel the treatment, but we at least need to know about it so that we know what precautions need to be taken. Depending on the illness that your child had, there may also be some inflammatory response that can be actually potentially dangerous if we are not aware of it in advance. Really important that you tell us about all the procedures, surgeries, however minor, whether it is taking the tonsils out, taking the appendix out, you had a tooth done under anesthesia. We need to know about that before we put your child through sedation or general anesthesia for administration of any intrathecal drugs, so ASO and other drugs. Medication. I cannot emphasize this enough. You have to tell us about every single medication that your child is taking or has been taking recently, because that can impact the potential efficacy and safety profile of the drugs, as well as the medication that may be used to sedate or to put your child under general anesthesia. And remember that over-the-counter medications that you get, even things like Tylenol, ibuprofen, they all have side effects. They can interact with the medications that we're giving. Supplements. A lot of people do not think about supplements, herbal supplements. They may be natural products. every supplement has the potential for side effects. We need to know about it. Again, it does not mean that your child will not qualify for the study at all, but we just need to know in advance that they are taking these supplements. If your child is on some cannabidiol treatment, Epidiolex, we need to know about it, right? You go out on over the counter or to some medical marijuana shop and you buy something for your kid, that's fine, but let us know about it. Really important when it comes to supplements and over-the-counter stuff that you tell us the exact brand and the dosage, because if you take the cannabis products, for example, that are out on the market, there are many different brands, as you know, and every brand has slightly different composition. So if any side effects would occur, we need to know what exactly your child was taking. And if there are changes in the dose or the dosing regimen, you've gone from twice a day to 3 times a day, or you've gone up from 5 milligrams to 7.5 milligrams, we need to know about these things again. I think the other thing I emphasize to parents from a safety perspective is we want to know about all side effects. It doesn't matter whether you think that side effect is due to the study medication that your child just received, because if you only tell us what you think is a side effect, we may be missing things. There may be things that are side effects that because they were not reported to us, we did not know about them. So really important that you tell us about everything and you let the clinicians, you let your study doctors decide whether or not to report them to the sponsor. So this is really just a cartoon to remind you that all medications, both prescription medications, over-the-counter medications, herbal supplements, cannabis, they all have side effects. There is not a single medication in this world without side effects and do not necessarily interact with some substance. So there's drug-drug interaction. It can interact with food, interact with other things. This is why we need to know about it. Really, really important. And it's not just an academic exercise, right? This is not just, well, we are curious, we want to know about it. If something were to happen, we have reported to FDA, this is a form that we have to use. And you will see that the FDA requires us as investigators to complete this form, which includes concomitant, i.e. what other products are you using, right? So we need to know name, we need to know when you started that product, when you stopped using that product, if you stopped using that product, and it's really detailed. So we have a legal obligation to actually find out from you, and you have a legal obligation to tell us that you know what medications you're on, because if you don't tell us what your child is taking, we can't tell the FDA that. So we need your help. We need you to tell us what your child is taking. All right, I'm going to hand this back to Dr. Barry Kravitz, who is going to tell you about other best practices.
Okay. I'm going to finish this off with a bit of a pep talk and then with a bit of a warning. Okay, so we know that being in a trial is a lot of work. This is not just coming to clinic. There are specific things you have to do, a lot of things, and your child needs more care than a typical child. So it's very hard. We understand, we get that. There's certain things that we have to do also. So the most important thing before you get into a trial is educate yourself on what is involved. We send you out the consent ahead of time. We give you lots of information. Can you do it? Can you get through everything? Are you okay with spinal taps if it's an ASO trial to deliver the drug into the spinal fluid? Are you okay with anesthesia? How long will the trial go on? Can you participate? Will you stay in the trial? Will you be able to do all the requirements? The requirements are much more than clinical visits. Everything has to be done exactly according to the protocol. We can't make exceptions, and we can't just do this because it would be more convenient. There are times when we may not agree with all the things we have to do, but we still have to do them. So except when needed for medical reasons, we can't make exceptions. And then in a lot of these trials, there are a lot of diaries and assessment forms and evaluations, and you really need to approach those very honestly. Don't embellish what your child can do, and also don't underrate what they can do. You really have to try to think hard about where they are exactly, and you have to do all these assessments. You have to do your videos on time, or use your devices when you're told to. If you don't do this, it may hurt the data. And if you're going to really have trouble getting through all of that, it's probably better for someone else to be in the trial to have the best chance of the drug being approved. So it's important to think about whether you can really do everything that is going to be, there's going to be some burden. We have to have some burden in order to get to the end and have these products approved. If participants pull out or just don't do all the activities required, we might need to recruit people to fill the space. We may have incomplete data. The FDA doesn't like incomplete data. And it may delay the trial finish if lots of patients drop out. So getting to completion is the goal. We want to minimize the people who will leave the trial because it's too stressful. So we want people to think about it ahead of time and educate yourself on what's involved. And if you realistically think you can do everything and do it well then, and you're all in and you're super motivated, we want you in the trial because we obviously need patients in the trial to get the drug approved. But once you're in, others are depending on you for their future treatment. You are really allowing the studies that will let your brethren that can't be in a trial get access to treatment in the future. So don't let them down. You're in partnership with us. We will do everything within our power to help you. Some things are out of our power, and with the study sponsor or the company that's running the trial. So this is a really important message, I think. But also, there's another thing that I really need to talk about. And the reason I need to talk about this is that we are going into phase 3 trials, which will be placebo controlled. And in fact, they won't be placebo controlled. They will be sham controlled, it looks like, at the dictation of the FDA. And what that means is that your study site is going to have a team that knows whether you're getting sham or drug. And there's going to be a team that doesn't know. And things that you post, things that you communicate could not only unblind other people, could not only produce placebo effect in other families, but they could unblind your study staff, which is one way to really, cause the trial to fail. So it's very important when you are in a trial not to post stuff publicly about your child's side effects or responses. Be in a Facebook group with other trial participants, compare effects in your child with others you know who are a child, go in the room at Rush and talk with each other about what's happening, compare what your kid is doing with somebody else. We can't have that. We had it in phase 2. Yes, we kind of didn't realize how much of it we would have. In phase 3, we cannot have this. You're not helping anyone, including yourself, if you do this, and it can cause trials to fail and then no one gets the drug. You can talk to your PI you will have rules in a sham controlled trial about who you can talk to about what and who you can't. You can post how much you love your trial site, where to find parking, your favorite coordinator, the best food at the hospital, how to get your safety bed set up in the room, what equipment the clinic has and what you need to bring. This is all helpful information that won't cause a problem with the trial. But we don't want to see the video of your child saying a new word or pointing out letters or your Fitbit output because you slept through the night, which means your kid slept through the night. That is stuff you can't post. And I'm just going to tell you a quick story at the end here about Fragile X syndrome, where we fell into a rabbit hole earlier in social media when we didn't realize what would happen. So we had a trial of a drug called arbaclofen, which failed in part because of placebo effects related to families. Families posted how to rate the scale to get into the trial, and then they would increase their ratings on the scale to be in the trial. And then, of course, as the trial went on, we had a huge placebo effect as their ratings came down to what the performance actually was. So we had this trial, and right there in the primary outcome, we had a huge placebo effect because of families gaming the system and boosting their scores and also posting stuff on Facebook about what their kid was doing, which raised our placebo effect. We had significant improvement in several measures that were secondary measures, but we can't get the drug approved with FDA because we failed on the primary measure. And this kind of thing can happen in any disorder, any neurodevelopmental disability. We have families with Fragile X who are still pining for this drug because it really worked for them. But we there was what happened during this trial did significantly contribute to our placebo effect and thus our lack of ability to have access to the drug. So you in the Angelman world do not want to be like the Fragile X families. And we've talked to them too. So do not post these things on social media. The top 10 things you should never post on social media are anything that's going on with your child in the trial. Okay, that's my warning. So let's not go down the same path that we know is not good. And so with that, we're going to have a question and answer session, and we will take questions. Oh, it looks like you have to come up and go to the microphone. Is that not on, Terry Jo?
Okay. I just want to say, first of all, thank you so much for doing these clinical trials. You know you're my hero in the world, and I appreciate all of you so much. So that's one thing, I think we should clap. And the second thing is, I just wanted to make two comments. One, I want, you know, so I can say that.
Thank you, Terry Jo. We're not trying to dissuade you from being in the trials. We just want you to enter it with a huge amount of energy and an understanding that you're going to have to do a lot of work. We're going to have to do a lot of work. You're going to have to do a lot of work. In the end, we're going to get over the finish line.
So sorry, I may not have been clear about that. So the strategy doesn't do anything to the maternal gene. The maternal gene is either not present because of a deletion or it's disabled because of a typo in it. So the strategy does nothing to the maternal gene. What it does is the ASO tricks the body's own mechanism, the RNase H mechanism, to chop apart the antisense transcript, which is keeping the paternal gene asleep. So we're going to take the repressor, we're going to stop the stop. So this gene is being kept asleep, inactive by this long antisense transcript. The antisense oligonucleotide is an antisense to the antisense. And so we're going to trigger the body to destroy that antisense transcript and allow the paternal gene to be expressed so that we can make some protein from it. Makes sense. We sure hope so. we want to be inclusive of everybody. We would like to be able to welcome in UPD and imprinting defect patients. Each sponsor has their own protocols which specify who can and cannot get in the trial. They're called inclusion exclusion criteria. The investigators don't have control over that, although we like to give them some input with our opinions. So it's going to be different for every protocol. But we hope that we are at a point now where we'll be able to start welcoming in the genotypes that we haven't been able to study up till now.
Those evaluations have been collected pretty much at the same time in all the trials. You get a dose of medication, and then right before you come in, sometimes not every time, but when you come in to get a dose, usually the evaluations are done before the dose. What this has led to is that sometimes the wait between doses has been long, and some of the effects may start to be wearing off. And so the evaluations at this point have picked up what we call trough improvements. In other words, if the patient got like more better in the first 2 or 3 months, and then they came in at 4 months for their assessment, and it might have been not quite as good. Probably the timing of those assessments will move for phase 3 trials, because now we understand this a little bit better. And the specific assessments we're using in Angelman syndrome, they've been used in trials before that the FDA has accepted. Has the primary outcome been the Bailey? I can't think of one where that was the case, but the FDA really needs to evolve at this point, because we're starting to treat a lot of genetic disorders where we need new outcomes that aren't standard. The CGI that Wenhan talked about has been used for approval in a large number of studies.
Okay. I just want to add one more thing. These outcome measures were chosen because we have a lot of natural history data out there. These have been used in the natural history study, which has been going on since 2006. And we have a lot of natural history data on these outcome measures of how do our individuals, without an intervention, without this ASO intervention, how are they doing on it? So they're going to be publishing, they're going to be showing data. They're going to show how their data compares to the natural history data. And so that's why these outcome measures were selected for these trials, because we know how our kids work on this measure without this intervention.
Good morning, everyone. Thank you to all our researchers. So my question is regarding the natural history study to Dr. Lynn Bird. We were part of the study for years until maybe we haven't gone back for at least 8 years now. So what is the chance of going? I mean, can we continue on with the study?
Yes, we want you back.
Okay. Okay. And then, so the natural history is.
You still have my email. I know you do.
Yeah, I do. So we live in Vegas, and is there any closer clinic that we can go to than Rady's Hospital?
Well, you're asking two different questions. So coming to clinic and coming to be part of the natural history study are two different things, but they often happen simultaneously. It's possible that there might be a closer clinic. I don't know of one, but there certainly is not a closer natural history site.
I got you.
Come see it.
Thank you. And a question on the ASO trials. At what phase are you in right now? And how many subjects do you need to complete the study? And what is the time frame from from start to finish? And where is it done? What clinic? Where do we go for this? Like the site location?
So there's not just one study going on. There's 3 different companies sponsoring 3 different trials. The phase 1/2 trials are all fully enrolled. There's nobody being enrolled now. They've all rolled over into a long-term extension phase. But the best place to go look to find out what trials are available and what sites are conducting those trials is the website www.clinicaltrials.gov. And so any trial that accepts federal funding must post on there, but many trials that are sponsored by industry also are found on that website because they know people go there to look.
Okay, thank you so much.